Welcome everyone to our talk today. I'm very happy to introduce Dr. Thomas Liu. He's a professor of radiology, psychiatry, and bioengineering at the University of California in San Diego. He's also the director of the Center for Functional MRI at UC San Diego. He received his PhD in electrical engineering from Stanford, where he worked on detecting ultra-low frequency magnetic fields in earthquake faults. Very interesting background. And since he joined UC San Diego in 1999, his research has spanned a broad range of areas in neuroimaging, including experimental design and analysis, signal processing, and modeling of the fMRI signal, as well as multimodal fMRI, EEG, and MEG imaging. He is a recipient of the Distinguished Investigator Award from the American Academy of Radiology Research and the Outstanding Teacher Award from the International Society for Magnetic Resonance in Medicine. It is a great privilege to have Dr. Thomas Liu here with us today and hear his talk about the controversies, hypotheses, and discoveries in resting state global brain activity. Dr. Liu, you have the floor. Thank you.
Great. Thank you, Rogers, for the very nice introduction. And thank you to the Brain Space Initiative for inviting me to give a talk in your seminar series. So today I'd like to give sort of an overview of some of the work that's being done looking at racing state global brain activity. So first, let me talk about, and in this specific, we'll be talking about what's the global signal in fMRI, and then maybe touch upon how it's related to other resting state activity. But the first question we want to look at is defining what the global signal is and why do we care about it. So most of you are familiar with resting state fMRI, where we measure brain activity without sort of any sort of specific stimulus. And resting state fMRI is now, widely prevalent. It's the field has discovered many different resting state networks, including this sort of landmark multimodal parcellation of the brain by the Human Connectome Project. Of all the sort of one of the more famous networks that people have talked about, obviously, is the default mode network. And that network sort of really was, sort of due mostly to the work of Marcus Raichle at Washington University, who sort of defined this default mode network, which has this property that basically deactivates during a task. And so this led to this sort of very famous paper in PNAS from 2005, looking at the default mode network here, where here we have the seed in the posterior cingulate cortex, and we're seeing the default mode network here in red. And what you're also seeing is there are areas that are in blue that are anti-correlated to the default mode network. And if you look at the time course, the default mode network, when it's going down, it seems like there's an anti-correlated network going up, and vice versa. And so this led to this sort of view of this sort of two opposing networks, perhaps the default mode network more active when you're sort of looking at your internal state, and then switching to another task positive or attention network when you're paying attention to the external environment. This is a very appealing view of the brain alternating between these two states. But that was followed by some work from Kevin Murphy from Peter Bannattini's group at the NIH, questioning whether these two anti-correlated networks really existed. So if they did something called global signal regression, then they indeed could find these two anti-correlated networks. But if they didn't do global signal regression, they really only found one strong positive network. And so this really led to this question about whether this fairly appealing picture of the brain alternating between states was really a true biological attribute, or was just an artifact of the processing. So before I go on, let me just define what the global signal is so everyone's on the same page. It's fairly simple. It's essentially just the mean signal over the brain at each time point. So for example, here I'm showing just one slice in the brain at different time points. And if I simply average all of the voxels in the brain at that time point, I would get its global signal value. So here where you see a lot of positive values in the brain, the global signal is red here, okay? And here where you see a lot of negative signals and the global signal is negative. And so it's really just sort of saying what is the average value of the brain at each time point. So um, now global signal regression means you take that signal, and so you compute the global signal as that average, and then for every voxel in the brain, you simply project it out using this operation here. And so you essentially you regress the global signal out of every time series in your brain, and then you proceed with your processing. So that process is known as global signal regression. Now, why do people do global signal regression? Well, why it's popular and why it gained sort of popular traction is that if you look at resting state functional connectivity maps using, say, very basic methods such as seed correlation. So, for example, if I put a seed voxel here in the posterior ligament sync cortex, in some subjects I may get lucky and I get a very sort of classic default mode network map. But in other subjects, we really don't see anything if you don't do any further processing. Now, there are alternatives, such as some of you may be regressing out white matter and CSF from your data. There are other methods called post hoc centering and median angle shift that are not really typically used. But if you look at what you get with global signal regression, it really does a very good job of recovering sort of these, what you would expect to see for a default mode network. And so it's just a very simple way of cleaning up the data. And that's really, one of the reasons why it's been adopted so widely. And so one of the advantages of doing things like global signal regression is, for example, if you're looking at the association between, say, functional connectivity behavior, this paper here found that if you did global signal regression, you'd get a much stronger association, sort of much more variance explained when you do global signal regression versus when you don't do global signal regression. So it does seem to clean up the data quite a bit for studies. But the controversy is really sort of summarized by this plot here, which is essentially, if you looked at, and each of these lines is the subject, and it's looking at the histogram of correlation values across the brain with the posterior ligament cortex, and you can sort of see that there's a real positive bias in the correlation values. Okay. Now, if you do global signal regression, it actually forces all these histograms to be centered around 0. And so regardless of what your initial histogram was, you essentially are forcing the existence of both positive and negative correlations. And this was the original argument Murphy made that potentially these anti-correlations were artifactual. From a geometric point of view, if you could think of sort of every time series in the brain as being represented by a multidimensional vector, and let's just say that we had two vectors like this, and this is its global signal, which is sort of the average of all these vectors, say, around a cone. If you project this out, then you are forced to have all your vectors lie in this plane. And so these vectors that had some positive correlation are now going to be anti-correlated. And so mathematically, you're imposing these anti-correlations. So this has led to these papers now already 7 years old, but Kevin Murphy and Michael Fox wrote a paper, and I also, our group wrote a paper about 7 years ago, sort of discussing the state of the art of what the global signal was there, and discussing, what were its implications. At the time, we felt we had a pretty good understanding of it, and that, there wasn't much more to be said. I mean, that there was clearly evidence that the global signal had both nuisance or information. But since then, there's probably even more renewed interest. Some of you may have been at the OHBM. There was a symposium there that I was part of where there were still sort of this discussion about the global signal and the debate about whether to use it and how it should be viewed in the fMRI field. And so I think that although we have made progress, certainly the debate is not resolved yet. So my goal is to sort of give you some more insight into some of what might be some of the issues and how this might affect how you think about your data. So the first question is, why don't we just not do global signal regression? I mean, if it's so controversial, why don't we just avoid it? This is a nice sort of review paper in Nature Protocols showing that, anyone who does neuroimaging knows that there's lots of different pre-processing options, and global signal regression is certainly one of those pre-processing options. but something that's become more popular is to use alternatives to it, such as, say, CompCore. Physiological nuisance regression is used quite a bit. Maybe you might use ICA. So there are lots of options. And so maybe one of the things you might think is, well, why don't we just use something like CompCore and we can get away from sort of the issues of global signal regression. And in fact, that is sort of what many groups are using now for resting state functional connectivity, is using some version of CompCore. So this is a paper looking at what's called anatomical CompCore. And they're saying that if you do global signal regression or CompCore, you can recover these anti-coordinate networks, but you're not doing global signal regression. And in fact, things like CompCore are parts of very standard processing pipelines, such as fMRI prep. So there is a high likelihood that even if you're not doing global signal regression, perhaps you're doing something like CompCore. Now, I sort of have a sort of vested interest in understanding whether this really makes sense, because it's actually my group, very talented graduate student who invented CompCore. And when we invented CompCore, not for resting state fMRI, but for task-related fMRI, where you knew what the task was. And so we could be very careful in terms of making sure that the CompCore components we pick from white matter CSF were not going to affect our analysis. Now, for resting state analysis, where there is no clear task, then it becomes a little more difficult to say whether what you're removing or not is what you should be removing. In addition, we were very careful to use very high thresholds for our partial voluming, whereas most implementation of CompCore use fairly loose thresholds for their partial voluming of white matter and CSF. And so my concern is that many people using CompCore are in fact including parts of the global signal in the CompCore regressors anyways. And so even if you think you're not doing global signal regression, you are doing some version of global signal regression. And in fact, if you look at sort of the components of the global signal, if you don't do any processing, this is what the global signal looks like. And if you take out white matter and CSF and all the other components, then you can sort of see that the white matter and CSF do make up a considerable part of the global signal. And so that if you're including any white matter and CSF components in your calculation of CompCore, then you probably are doing some global signal regression. What's more even interesting is, even if you're not doing CompCore and you're just doing sort of, let's say, motion regression, like taking the regressors and trying to regress out motion. This is a recent paper that we published in Imaging Neuroscience showing that, in fact, if you look at, for example, your motion estimates, so this is from a multi-echo scan where we have this motion estimates from the first echo in blue and from the second echo in green, and you look at the difference in those motion estimates, you get this signal. And this difference in motion estimates is essentially the global signal. Okay, so it turns out many image reconstruction algorithms, because they're image-based, will pick up global resting state activity. And so that, especially in the second echo, where there's more bold weighting, you will have a global signal component in your motion regressor. And therefore, even doing something as apparently benign as doing motion regression can actually do some form of global signal regression. Okay, so basically the point is that global signal regression is probably most pipelines are doing some sort of global signal regression. The next thing I want to talk about is sort of get a little deeper into sort of what are some of the characteristics of the global signal. So one thing is that the global signal is one of those things that it's not just the mean waveform in the brain, but it's just something that it's like everywhere you look, you're probably going to see some signature of the global signal. So this is from an abstract that one of my visiting graduate students did. So looking at the global signal here, but then also just looking at sort of the signal in either some of the draining veins, and you can sort of see very strong correlation between the global signal and the signal in the draining veins. This is showing the global signal here in blue from a paper by Erdogan and Tong and others. And then here are essentially in green are measures of oxyhemoglobin measured at either the fingertip, measured at the fingertip. And so even if you measure take measurements of oxygenation at either the fingertip, I think they even did this at the toe, you will see signatures of the global signal. So really, this is even more global than the brain. And then in terms of how you might think about what's happening with the global signal, uh, an important thing to think about is that even though it is just a mathematical construct where we're taking the average of the brain images, if you look at the individual images, which is generally a good thing to do, is just look at the data as it is, you can sort of see that already there's very strong, when the global signal is high, there's a lot of spatial homogeneity in the brain image. Here, very negative, here, very positive. And you can see when the global signal is very low, you're already seeing these anti-correlations in individual time points. And so this idea that the global signal has this sort of time characteristics where you might want to take that into account when looking at the global signal. And in fact, this is very much related to this concept of co-activation patterns from Xiaolu and Jeff Dunn at the NIH, where they found that when we do these correlation maps, if you do a typical correlation map, you get this. But if you simply take the time average of points that are sort of say correlate, where the PCC signal is high, you get something very similar. And so the idea is that correlation tends to be dominated by points in the time series that are very high, and that's what determines your correlation. So what that means is that for a global signal, it means that if you just ignored those points in time where the global signal was high and just kept all the other remaining time points and looked at that, then essentially you could just get a map that looks very much like what you would get with global signal regression. And this anti-correlation in the time points is real. It's not a mathematical artifact because basically you're looking at the correlation, the functional connectivity in those time points where the global signal was just had a low value. So that's not surprising given the data we showed in the previous slide, but just the idea that when we think about the global signal, it's useful to think about it as being sort of dominated by these very large excursions of positive and negative values. And in between, there might be sort of information that we can see through in terms of the minimizing the effects of the global signal. Now, I think there's been also an emerging view from several groups, including the one at Penn State, and this is also a group from, I believe, partly from Sheila Kelpolz's lab and also Lucinda Adin's lab. So this idea that the global signal is really related to propagation of signals, either bottom up, going from sensor areas to high order areas, or top down, going from high order areas to sensor areas, and that there's a classic sort of propagation along the principal gradient direction of the signal that seems to happen when the global signal is making a transition. And in this work, using a complex principal component analysis, they're able to sort of track sort of the wave propagation related to the global signal. And so the idea is that an emerging view of the global signal may not just be viewing it as a signal in time, but really we need to view it as sort of the space-time signal that's giving these that's occurring and giving these signal fluctuations. Interestingly enough, we don't just see this in animals, in humans, and it's not just a vascular artifact. This is some nice work done using data from the Human Brain Atlas and finding that there are similar global signal propagations that occur using sort of neuropixel measurements. And so it really isn't just a neurovascular confound specific to fMRI. It really does seem to be something that's happening in the brain, where the brain does seem to be set up to have these global ways of propagation. The next thing I want to talk about is that I think when the global signal first emerged, it was very much considered a nuisance term, but over the last 10 years or so, I think there's really been this interesting use of the global signal as more of a source of information. So I'd like to go over some of that. Probably the first one was this paper here in PNAS showing that in schizophrenia, there is higher global signal in the schizophrenic patients versus the healthy controls. In this paper, there was sort of this very sort of sort of lengthy and complex interpretation of this. We'll come back to this maybe later in terms of why this global signal might be higher in the schizophrenics versus the healthy controls, but sort of the first use of the perhaps a global signal is an independent metric that you could use to differentiate populations. This interesting paper showing that basically looking at visual detection as a function of time of day and your ability to detect things is much better early in the day when and later sort of near sunset and near dawn when you sort of have to be basically able to detect things when the light is not so good. And you can sort of see they're finding the global signal, the mean global signal is lower at those points in the day. This is a paper looking at mind blanking and showing that when your mind blanks out, it tends to be that your global signal amplitude is higher when you sort of have these mind blanking incidents. There's also this emerging area of looking at what's called global signal topography, which is simply you take the global signal and you correlate it with every voxel in the brain, sort of a several double use of the data, but basically areas here in red are showing where global signal is high, so the yellow is the highest parts of very strong global signal components, and the red tends to be lower global signal components. And what they're able to do now then is to look at differences in topography, for example, in bipolar disorder, looking at differences in the topography of the global signal versus controls. And so using sort of this additional global signal property. Another interesting use of the global signal, this is the sort of very seminal paper by the group from MGH looking at CSF flow, which is becoming of great interest now, especially the lymphatic system and sort of how well the brain can sort of flush out toxins. And so here, what they're showing is if you take the global signal here in green, and it turns out that if you look at where the ventricle is flushing through the CSF is flushing through the fourth ventricle, it turns out it's really where these derivatives in the global signal are happening. And so, for example, if you take the derivative of this green signal, so the DDT bold, it's basically right, it explains very well when the CSF is being sort of pumped through the fourth ventricle. And so this idea is that this signal, which seems to be just a measure of brain activity, is also somehow giving you a sense of how the brain is actually mechanically pulsating in time in conjunction with these spurts of CSF flow. And finally, some very interesting work just published last year in Nature Communications, showing that basically, if you look at the global signal, it tells you a lot about where beta amyloid is being collected in the brain. And if the global signal is low, or the propagation of the global signal is not very strong, then you will tend to have more beta amyloid collection than where the global signal is sort of having is stronger. And so there are implications here, once again, that this measure of how the brain is acting in a global fashion, it can give you insight into other disease states and populations. Okay, so now let me switch gears a little bit and talk about what drives the global signal. So I'm just going to give some a view from data from our lab. Obviously, there's other work, but in our lab, sort of one of the insights we had in global signal regression came from this study where we looked at subjects before and after a dose of caffeine. And so if you looked at the PCC maps before caffeine, they tend to look like this. If you did global signal regression, then we see these anti-correlating networks showing up. But what we noticed is after caffeine, it's not quite the same as global signal regression, but it does quite a good job of sort of getting rid of these sort of very positive correlations, and we start to see these anti-correlations appearing, and it's a little more similar. These two are arguably more similar than these two, okay? So that, in a sense, the caffeine is sort of what we call sort of doing some sort of natural global signal regression and doing something in the physiology is reducing a global signal component. And in fact, if we actually measure the global signal amplitude, which is sort of simply the variance of the global signal, what we find is that there is a significant reduction in that global signal amplitude with the caffeine dose. Now, at the time, we really weren't sure if this was a vascular effect or a neural effect, because caffeine not only, as everyone knows, sort of is a neurostimulant, but it also reduces your blood flow by quite a lot. So, for example, a 200 milligram dose of caffeine, which is what we use in this studies, typically reduces blood flow by about 30%. Okay, so given that the bold signal is sort of a complex function of cerebral blood flow, blood volume, metabolism, we felt we needed some neuroelectrical measurements to sort of see what was really driving this effect. So one of the things we did was we used simultaneous EEG fMRI, which involves measuring the EEG waveforms within the scanner. That the EEG data is quite noisy when you do that, but with sort of modern methods of cleaning up the data, you can get to the point where there is meaningful data. And for this study, we sort of settled on a measure of what we call vigilance. And this is simply the alpha power divided by the delta and theta power, such that when alpha goes up, you tend to be more vigilant. And conversely, when delta and theta go up, that indicates you're becoming more drowsy. This is also known as the alpha slow wave index in the literature. Okay, so let's take a look at some of the data. So the interesting thing about this data that took us quite a while to sort of parse out was that not all subjects had the same effect. So for example, in this subject here, if you look at their EEG spectra, they're quite similar pre and post dose. And if you look at their resting state maps, they're also quite similar. So in this case, the global signal will be quite similar between these two states. On the other hand, other subjects, if you look at their EEG spectra, are quite different. And so pre-dose, you sort of see this green spectrum, and then post-dose, you see an increase, especially around the alpha band, indicating there is a sort of very pronounced increase in their vigilance level because the alpha power is going up. And in fact, if you look at their resting state map before the caffeine, you sort of see a lot of positive correlations. And then if you look at it after caffeine, you start to see these anti-correlations showing up. Okay, so there's a very strong difference in the global signal component between these two states. And so what we can do is for every subject, we can look at the change in their vigilance with the caffeine dose and also the change in their global signal amplitude. And what we find is this very striking relationship between the change in the global signal amplitude and the change in the vigilance. And this is, in this case, these are due to caffeine. And so here, the intersubjective variability turned out to be sort of, at first, we were sort of disappointed that some subjects didn't show an effect, but then we found out that we could use that to map out this intersubject variability quite nicely. In the same study, we also looked at whether the subjects had their eyes open or closed. And so, for example, if the eyes are closed, you sort of see versus in some subjects, we saw that there's almost no difference whether they open or close their eyes in the EEG spectrum. And in other subjects, similar to what we saw with caffeine, there's a large effect. And so there's a lot of intersubjective variability in how people's vigilance changes, even with simple manipulations such as opening and closing their eyes. Once again, we could look at the change in global signal amplitude and the change in vigilance. We got this very nice relationship here. And even more interesting that we found that if we mapped the difference in global signal amplitude versus vigilance for the caffeine data and the eyes open, eyes closed data, we found very similar relationships, indicating that perhaps it's really not how you modulate the vigilance that matters, but just the fact that the vigilance is changing, that's driving the change in global signal amplitude. Furthermore, you can look at the sort of the natural variability in subjects before caffeine. And so this is essentially looking at 3 different subjects. And so, for example, this subject here, who has very high vigilance, you can sort of see their map looks like that. So then if we go to subjects sort of with medium vigilance, we see something like this. And then with drowse, this is the drowsy subject, which is such much higher global signal. And so you're sort of seeing across subjects, we can look at their global signal amplitude and their vigilance, and we're seeing this sort of effect across the population. Now, there has been similar work done. This is a nice study using data from the HCP, looking at the global signal amplitude as a function of the time throughout the 15-minute HCP resting state scan. And as you can see, the global signal amplitude is steadily rising as they're going through the scan. And given what we just saw, that means that when the global signal amplitude is increasing, their vigilance level is decreasing quite a bit. And that's very consistent with other data in the field showing that typically after about 5 minutes, most subjects are asleep in the scanner in a resting state protocol, indicating very low vigilance levels. And so that's something definitely to keep in mind as you're analyzing these data sets, especially any long resting state scans from these from some the large public databases that are available now. The other thing to realize is that these changes, this relationship between the vigilance and global signal actually happens across time. And so, for example, with the EEG fMRI, we can, for every time point, we can measure not only the global signal, but we can also measure the vigilance. And here I'm showing the global signal inverted in red and the vigilance, and you can sort of see a very strong correlation in the two signals, indicating that even over the course of a time series, whenever the vigilance tends to be high, if you actually just look at the data itself, you see these patterns in the data. These are just sort of average of the time, the images at the points of high vigilance. And then if you average the data at points of low vigilance, you see these. So there just tends to be higher fMRI signal patterns of higher global fMRI signal when there's low vigilance, and there tends to be more negative values when there's high vigilance. And so that's something to keep in mind. This also matches with the fact that if you look at where the global signal is going up in time, using sort of electrophysiological data, you can actually see that there are these sort of very stereotypical changes in the EEG sort of time varying spectrum, indicating that there are, whatever the brain's doing when it's making the global signal go into either high state or low state, there is something that is also occurring in the EEG signature. We see this not only in humans, but in mice. So for example, here, if we use pupil as an indicator of how vigilant they are, you can sort of see that there is going to be this sort of this predicted change as they become more vigilant, the global signal goes down, and this is across time as well. So what we can do is we can sort of use that insight of the fact that the vigilance is varying across the time series to come back and get a better understanding of what global signal regression might be doing. So for example, we've just said that when the global signal is low, we tend to see these sort of patterns in the data. When the global signal is very negative, or sorry, when the global signal amplitude is small, close to zero, we can see these patterns in the data. When the global signal is very negative, we see very negative values in the data. And when it's very positive, we see very positive values in the data. And we know that what we just said is that when it's low, this tends to be areas of high vigilance, and this tends to be areas of very low vigilance. And so if we just retain the points where the global signal magnitude is low, then we see anti-correlations. And essentially what we're doing is we're censoring out all those time points where the vigilance was either going to a high value or a low value. And so in a sense that when we're doing things like global signal regression or things like global signal censoring, essentially we are attenuating the effects of vigilance variations. And so whether this is a good thing or a bad thing will depend on your study and the design, because if you're trying to minimize, if you don't really care about differences in vigilance variations, then you might want to do global signal regression. But if you think that that might be carrying information about your state or population, then doing global signal regression might remove that information from your analysis. So for example, in this study, looking at default dynamic functional connectivity, what they find is if you didn't do global signal regression, you can find these different states that look, there are differences in these states. But if you do global signal regression, all of a sudden these states all look fairly much more similar to each other. And so that the global signal regression is potentially reducing any vigilance-related fluctuations in your dynamic functional connectivity states. And finally, just one to relate this back to some of the data we talked about, where we were looking at waves, either going bottom up or top down. What this group found was that if you looked at how drowsy subjects were, okay, whether they were high, medium, or low drowsiness, it turns out that the ratio of these top-down propagations varied. And so, for example, when subjects were not very drowsy, then you got to see that there was much more top-down propagations versus when they became more drowsy. And so essentially, signal propagating from the higher order areas down to the lower areas seems to be enhanced when you're less drowsy, and then it decreases as you become more drowsy. And interesting enough, they found this both in humans and in monkeys, indicating that there is something that's conserved across species in how the global signal operates. Okay, so then let me just end with some thoughts about what this means and what are the implications and how this ties into how we think about fMRI data and neuroimaging analysis in general. I think one thing that is sort of some recent papers have been sort of emphasizing is that although typically many fMRI scans just measure the fMRI signal, that there is increasing interest in measuring other things such as pupil diameter, respiratory volume, heart rate variability, peripheral blood vessel tone, maybe EEG if possible. And it turns out that when you do these types of measurements, almost everything that you can measure is correlated to the global signal. Okay. And this is another paper showing that as the global signal is changing, there are lots of things happening at the same time, such as differences in respiratory volume, heart rate reduction, CSF inflow. So essentially anything you can measure about the brain seems to be related to everything else. These are just some examples where we're looking at the cross correlation between global signal is sort of this principal component one, looking at how it's correlated to respiratory volume, heart rate, CSF fluctuations. So really, the idea is we can't really isolate any one aspect of what's happening with these global fluctuations. Now, this may not be too surprising if you think about, what the brain is designed to do. I mean, if you think that the brain, from an evolutionary point of view, really, you need the brain in order to make decisions and move your body and to survive. And so it does make sense that there must be some sort of global coordination of activity. And in fact, that most of the time you would expect the whole brain to be moving. And so, for example, in this study from NeuroPixel study from mice, you're seeing neural activity in 500 neurons, and you're seeing activity in basically widespread activity in all the neurons, even when the mouse is just sort of sitting there. And what they found in this study was if they then took images of the mouse, sort of just sitting there, but just sort of, moving its face and twitching and doing things like that, then that they found that they could actually predict some bit of the neural activity from just looking at the spontaneous behavior of the mouse as it's just sitting there. Okay. So I think an important thing to keep in mind as we're sort of progressing in our analysis of fMRI studies is that, even though we might want to just focus on the brain and get our nice fMRI data, that really there's so much going on here. And if we're not measuring all these things, it may be difficult to really understand what's going on in this part of the system, And this, I think, it behooves us to remember this sort of the law of the instrument that, typically we have this great tool of fMRI, and so we study what we can measure, and that leads to bias in ignoring sort of everything else that might be affecting the signal. I think the problem is even more going to be challenging as we move forward. We are in this day and age where we are lucky enough to have large data sets, open data sets. We also have new ways of looking at the data with AI and machine learning. And so with lots of data, it just means we're going to be able to come up with lots of different things to measure. And if we look at the data long enough, anyone who looks at fMRI data long enough, you will always get a result. And so the idea is we need to be careful about making sure we don't just start coming up with more and more unrelated results. And it really is, I think, behooves us as a field to start thinking about how are all these things tied together, as opposed to just coming up with another metric or another way of analyzing the data. And finally, I hope I've convinced you that the global signal is probably present in all of your data, and it's present, and whether you think you're getting rid of it or not. And depending on what you think about the global signal, it may affect how you interpret your data in terms of especially whether how you're dealing with changes in arousal or vigilance across your population or conditions. So if you think of sort of the brain as being this elephant, or the global signal as being this elephant, obviously, if you only have one view of it, you might interpret it as being a tree or a rope or a wall. If this is a global signal, in some cases, you might think of it as a nuisance term, or you might think of it as information, or you might think of it as some combination of the two. And so I think it is important to keep in mind that we still don't really have a good grasp of what we're trying to measure in the brain. And I think keeping some awareness of the global signal effects and global resting state activity effects may be important for sort of how you look at your data. So with that, I think I'm ahead of schedule, so that's probably a good time to end. And so I'd like to thank all the students and staff who contribute to this and the funding sources. Thank you for your attention, and happy to answer questions.
All right. Thank you, Dr. Liu. Very interesting talk. Now we are gonna move to the Q&A session. I'll start with the questions on chat. First one is from Cyrus. He's saying great talk. And he's asking, may drowsy drugs, for example, Benadryl, have opposite effects on brain signal or on global signal compared to caffeine?
Yeah, that's a good question. I mean, I think in general, I'm not sure if there's any studies specific looking at things like Benadryl, but certainly, given the data we have, anything that makes you less vigilant would tend to make the global signal higher, would be my prediction.
Mm-hmm. Yeah, I think you showed some results with just measures from the scanner itself, like drowsiness while in the scanner, right? That had
but those are just sort of without a drug, just sort of inherent
yeah,
drowsiness.
exactly, yeah.
Yes, yeah.
So yeah, I agree. Supporting evidence for that. So next one, also from Cyrus. May the global signal be related with tasks such as button presses?
That's a good question. I mean, I think certainly if you have a task-related paradigm, depending on how much of the brain it lights up, that will certainly be included in your global signal. Now, the nice thing with task related, obviously, is what the paradigm is. And so then, therefore, you can sort of say how much of that global signal activation is related to the task. Now, I didn't go over it in this paper, but in fact, global signal regression was first looked at for task-based fMRI. And it was found that if you did global signal regression, you would get almost completely different results than if you didn't do global signal regression, because there is I think the bottom line is the global signal is such a huge resting state background fluctuation that it sort of actually in some cases swamps the task related activations, especially if the subject is drowsy. And there are studies showing that, for example, where you are in the global signal, when you do the task affects how well you respond to your task. So your response times will also vary. So for example, because if you think about it, which makes sense, if it really is giving you some metric of your time varying vigilance and arousal, then if you do the task when you're not as alert, you're going to have an effect. So I think it is something that needs to be kept in mind in task related, but the fact of the matter is with task related, because you have you can set up your general linear model in a sort of a more direct fashion. Um, it is considered sort of if you're doing it correctly, then at least you can sort of say what variance is assigned to my task, and then the global signal becomes just sort of a source of variance. Now, if you regress it out, then you have a lot of issues, which I think
Mhm.
most analyses tend not to do that. Now, the caveat is if you're doing something like CompCore and you're not actually doing it correctly, then you might actually be doing some of the analysis incorrectly. But I think that's leads to a much bigger problem. It's just a bigger problem in the field of fMRI, which is that many people are using pipelines without really knowing what they're doing and all the pros and cons. And essentially, if you think about global signal regression, is literally just one line and maybe thousands of lines of code, and yet it could have a profound effect.
Mhm. Yeah, that's a very good observation. Um, Good. I had a question for myself, and that was if on a particular study vigilance is important, then do you think that we should not remove the global signal?
Yeah, I mean, I think that's something where it's hard to say have a one size fits all prescription. I think
Mhm.
it's just more the knowledge of it is then allows you to make the judgment, and therefore, obviously one trend that has been happening in publications is sometimes reviewers will ask you to do the analysis with and without global signal regression, right? And so
Mhm.
therefore, you would need to sort of explain that. And then obviously, for example, in the schizophrenia study that I mentioned, uh, they found that global signal was higher in the schizophrenic population, and there was some sort of fairly sort of involved sort of dynamical analysis of that and explanation for that. But a potentially simpler explanation could simply because it's known that vigilance tends to be lower in subjects with schizophrenia, and so it's not surprising that the global signal would be higher. So I guess the question is, you know, if you know that vigilance changes drive it, then vigilance is relatively easy to monitor. You don't need a $3 million scanner to tell you that one subject group is less vigilant than another subject group. So I
Mhm.
think I don't want to prescribe anything one size fits all, but it's just good to keep in mind that this is a fairly, I would say, significant source of variance in fMRI data. And maybe your method takes care of it, or maybe it doesn't. But just to be aware of it gives you a better chance of making analysis. Now that said, one of the issues we face in neuroimaging, especially resting state, is that um, it is possible almost to find that what you measure is correlated with something else that you care about, and so um, there's very little. It's very difficult for us to check whether the story is correct or not. And so, as long as the story is interesting and compelling, then sometimes, um, you know, there is this bias to just publish the result. And so we do need to be sort of cognizant that there may be other interpretations of the data as well.
Right, right. And I wanted to follow up on that idea of, because it was my next question, running the analysis both ways. Do you have any recommendations or guidelines on how to compare those results and determine uh, if and draw conclusions from it, basically? How what, is there any guideline for that?
Yeah, I mean, I don't think there is a guideline right now. I think my suggestion would be to sort of probably see if there is a difference in global signal between your conditions or amplitudes, and then if there is, um, then you can run the analysis both ways and see if um, the effective GSR is because it's removing the vigilance variations. So I think that's where additional measures are useful, because fMRI alone is really too unconstrained, right? And for example, that's one of the reasons we delved into multimodality imaging is we realized that there's just too many things that can change the fMRI signal, especially once you go away from healthy populations, right, into disease populations, older populations, where changes in the vascular state can have a profound effect on the fMRI signal. And therefore, if all you have is the fMRI signal, then it's very difficult to tell what's going on. So I think as opposed to one prescription, I would just argue that we probably just need more sort of confirmatory data or getting a fuller picture. In the same way that when we just measure the brain alone, we're missing out on what the body is doing, right? And so then basically, it's like we're measuring the tail of the elephant and trying to make a story about what the elephant is. And so I do think that although it will be a very difficult task, I do think that we do have to be careful that, I mean, fMRI is such a powerful tool that we think it can do everything, and yet we probably should be a little more cautious in proceeding.
Thank you. All right. Following on with the questions, may you give short information about what's the difference between the global signal regression and censoring?
Yeah, so global signal regression is typically what's done in most pipelines, which is simply you regress the global signal out of each voxel. Censoring is sort of an approach that we sort of developed to more understand what the global signal is doing or global signal regression is doing. And it comes down to the idea that when you do regression, there's this thing called the leveraging effect, which is any regression or any linear fit tends to be driven by the highest data points, right? Which everyone knows. But what that means is that when you do global signal regression, it's really going to be determined by the highest data points, so the positive and negative things. And so what it does is it tends to just then get rid of those big excursions in your data and leaves sort of the data where the global signal is not very high. So then in the limit, you could just say, well, let me just censor out the time points where the global signal is either very high or very low, or very negative or very positive. And so it's just a different way of realizing that in the limit, regression can become censoring, and that if you censor data, then the data you're left with is just the data in those time bins, and there is no mathematical problem with that. If you look at the correlation in the data that's left over, that is the correlation that was going on in those time points. And so I think that tends to be the conclusion there, is that because that exists with the very, sort of with censoring, that then these anti-correlations probably do exist in the brain. They're not just an artifact of the regression.
Got it. Thank you. Another question from Cyrus is MRI, and I think he means functional MRI, really the best way to measure global signal? Because it may not be an absolute measure and it may drift, or would something like PET FDG be better, which is absolute? Or do you think that fMRI drift really is an effect of global signal?
I think that the idea is obviously the fMRI has scanner drift and things are at play, but typically scanner drift you can sort of fairly well sort of describe with fairly low order Fourier terms or something like that. And so I think that the frequency of the global signal fluctuations is an area where it's physiological and has been shown to be related to things like EEG and MEG. And so we have enough data out there to say that while some of the fMRI signal could be, not related to neuroactivity, enough of it is that, I don't think that part is of concern. Certainly FDG PET would have its advantages, but obviously it has its huge disadvantage. It's not practical to do and very expensive. Now you could do something like arterial spin labeling, which tends to be much more immune to low frequency drifts. And so I do, there certainly are global signal effects there. But I don't, once again, it's just a lower SNR measurement. And so I think the fact of the matter is most of the data we have to deal with is fMRI data because it is such high SNR. And so it behooves us to understand what is in that data.
Got it. I think one of the points that relates to this question is I think you showed the result of some study, I think from Oxford, that showed that over time subjects in the scanner are less vigilant, and maybe that's what drove this question because it kind of looks like drift, right? That it's very low frequency, it seemed, in that particular result. So is that one of the examples of where the physiological changes might be mixed up with drift? Yeah, I think this is the
Yeah,
one.
I mean, I think the idea is that I believe they've already, I don't remember the details of this paper. I believe they've already probably projected out a lot of these terms. And I think the amplitude here is, my guess is it's probably some measure of the standard deviation or something. I mean, it's not just the signal itself. So yeah, it's not like you just measure the fMRI signal on its own. It's more just measuring, as we saw, it's really the variance in the signal that matters. And so I don't remember the details of that, but that would be my guess as what they're measuring. But yeah, I mean, I think that certainly, as we know, the devil is always in the details on how the analysis was done. But obviously, if I were going to do this analysis, then I would certainly take out, first you project out all the low frequency drifts, compute the global signal, and then what you're looking at is sort of the time varying variance. You know, so basically, are the fluctuation levels getting bigger over time?
Mhm.
And those fluctuations are not at a low frequency time scale.
Got it. Got it. Thank you. That makes sense. We are right on time, but I don't know, Armin, should we go for one more question
Sure.
or open for?
I think that's a good idea. There's a question from Daniel, if you don't mind to share.
Okay, let's see. This is a follow-up to, I guess, one of the questions I made. Perhaps the best way is to regress out the usual confounds, respiratory, HRV, head motion, etc., and compare to regressing these and global signal, thereby revealing the components of global signal that are not common to the usual regressors. And thus, that could be the information carrying portion of the global signal.
Yeah, I mean that's certainly you know, one way to look at it. But I think one of the issues is that even things like, you know, what I think this sort of divide we make between what is a confound and what is not is also something, even with something like respiratory variability, right? That is related to arousal state, and so whether it's a confound or not depends on what you think is a confound. Or heart rate variability is also related. It's telling you something about what the brain's doing, because I think I probably didn't explain it very well enough, but I think the main point of this figure is that all these things that we treat as confounds are really reflecting the brain state, right? And the brain's controlling your breathing, it's controlling your heart rate, and so it's a circular thing, right? It's like to say something is a confound in this means you draw a line and you say something, and yet so it's a challenge we have of how do you analyze a holistic system with sort of, you know, the tools we have, which are simply saying this is good, this is bad. And so, you know, ultimately probably the challenge, which may or may not be achievable, is that, you know, what we really need is a model, right, of all this. And then, but it's going to be fairly complicated and need lots of data, and but maybe one of you guys can figure it out.
Yeah. I think it might be a bit related with this idea of looking at systems. There's a question here about whether you think that ICA, independent component analysis, could address any of these issues or tackle them by regressing out the global signal.
Yeah, so that's a good question. I think you know, certainly there has been some work on looking at ICA, and I'm not. You guys are much more expert in ICA than I am, but my understanding is, especially spatial ICA, will try to take that global signal and, you know, parse it into, you know, different components, and so my understanding is that ICA doesn't really do a good job of getting rid of the global signal components, because it sort of puts it splits it up, but it's still left in the different components. And so if you reassemble them, you'll see there. Now, there was some work by Glasser and others at WashU showing that maybe there might be temporal IC approaches to dealing with global signal, but my understanding is that included analysis where you had to concatenate lots of data and may or may not be practical for most studies. So I do think it's something that we should be aware of. The tricky thing, obviously, is once you do something with ICA, then it's sometimes the interpretation can be more difficult, and it may not be the same across populations. And until you actually look at maybe populations with different very levels of vigilance and things like that, there might be something where it works in one population but not another. So I think that's certainly an interesting area of research.
Thank you.